So thanks, Ariel. And before we move on, we all know that in order to produce good research, we need a good administration. So I also want to thank Yael, who was had major contribution for the center over the years. So thank you, Yael. And our next presentation is by Roza Izgilov from Professor Dafna Benayahu's lab from the Department of Cell and Developmental Biology from the Faculty of Medical and Health Sciences. And the title of the talk is Advanced Glycation End Products, AGEs or AGEs, alter tissue stiffness and lead to metabolic dysfunction.
So hi everyone. I'm very honored to be here and present today. I present today a topic of my PhD project, an advanced glycation end product and how it alters the stiffness and lead to some metabolic dysfunction. So as all of us know, diabetes is a chronic metabolic disease that rises through the years. Everyone is familiar or know at least one person that diagnosed with diabetes. This is a disease that highly associated with overweight and obesity, when one of the main metabolic tissue in it is the adipose tissue and the adipocyte itself. In the diabetic, just a second. In diabetes, we have hyperglycemic situation that cause an elevated glucose in the blood and elevated AGEs levels. Who is those AGEs? The most known one is the hemoglobin A1C that serves actually as a biomarker to diagnose and maintain diabetic treatment. And these AGEs are created non-enzymatically when glucose or glucose derivatives as methylglyoxal, glycated protein, lipids, and even nucleic acid, and create this end product, glycated end product. So this glycation is occurred not only by glucose, as I mentioned, and can accelerate through different metabolic processes in our body, but is very strengthened through the diabetic situation and through the disease, and cause irreversible protein crosslink. But what is the role of those AGEs in diabetes? Is it one of the factors for the disease or just some byproduct that going together with the disease ongoing? So through the years, we see more and more studies that associate AGEs high level with different diabetic complications. But we still, the studies about the adipose tissue is still poorly investigated, and we don't really know if this is the cause to the disease and the pathology that we see. Through the recent years, we studied this glycation through different models, in vitro models, and we showed that the glycation by MGO or other components create structural changes in protein, create cross-linking and aggregation, and even elevate stiffness in treated adipocytes. If we will follow the previous session, we also showed recently a novel protein that can aggregate outside of the neurogenic diseases. Okay. So for this project, we wanted to our question was, our hypothesis was, if is those AGEs will create similar phenotype as in diabetes itself, and in this way we wanted to show, understand if it is a cause to the pathology. We used two different model systems, an in vitro adipocyte and a mice nutritional model. So for start, we treated the cultured adipocyte with methylglyoxal, and we saw that the treatment with methylglyoxal and AGEs creation interfere and decrease the ability of the adipocyte to differentiate properly, and decrease also adipogenic markers. After that, we wanted to understand why. Adipocytes is highly maintained through, and they really need a proper glucose intake. So we went to check all the system of the glucose transporting to the cells and the insulin signaling, and we saw that indeed we have, after a treatment, a decreased insulin signaling, and a disrupt glucose uptake to the cells by two different glucose transporters that interrupt through the methylglyoxal treatment. So after we saw that methylglyoxal and AGEs indeed interfere with some metabolic response in our cells, we wanted to understand what is happening in the mice model. So we created a mice model in which we treated mice 15 weeks with the MGO, and then we checked for the metabolic phenotype in the end. And we saw, and the results are strengthened, was going on together with our hypothesis. We get a diabetes-like phenotype in our AGE model when we had elevated fasting glucose and elevating peak after a glucose tolerance test, and to assess the insulin resistance of the mice, we also performed an insulin tolerance test that confirmed it. And in addition, our mice also had some weight gain elevation. So in this point, we wanted to understand what is causing to this phenotype, what the AGE is affecting on, and particularly with a focus on the adipose tissue of the mice. So we took the adipose tissue from control and treated mice to proteomic analysis, and we received an elevation and change in different proteins that correlated with extracellular metrics. This was a very interesting result because, as we know, through diabetic pathology, we don't have only the metabolic changes in the body, but we also have a lot of ECM changes and the fibrotic-like state. So it gave us a clue, maybe the AGE's effect on the body through the fibrosis pathway. So to assess that, we went to the adipose tissue to look for the changes in the ECM compartment, and also to some biomarkers that tell us if it is causing some stiffness changes. This we looked through activation of YAP1 protein that usually serves as a mechanosensing protein, and when the ECM or the cellular niche became stiffer, as in fibrotic situation, we have a translocation of YAP1 from the cytoplasm to the nucleus. So here's the results from the adipose tissue, and we can see here for start the image of the ECM itself. We can see that after a treatment, we indeed have more enlarged ECM content and higher YAP1 in the system that telling us about the stiffness elevation in the adipocytes niche. We went back to our cellular model to check if those phenotypes also happens in our cultured adipocytes, and indeed we can see that in control and MGO treated adipocyte, the ECM became more aggregated and cross-linked, and if we will go to the molecular signaling, YAP1 is beautifully translocates from the cytoplasm of the cell to the nuclei to the nucleus to indicate this surrounding changes of the stiffness. After we checked this point and understand that our model indeed received metabolic changes and ECM changes that related to fibrotic phenotype, we wanted to go further. As we know from the literature, changes in the ECM stiffness also correlates with cytoskeleton reorganization and nuclear changes. So we went to our model and to check this, and we can see that after MGO treatment, our adipocyte is entirely, the cytoskeleton of the adipocyte is entirely disrupted, and we had enlarged nucleus with enlarged nucleoli that correlates with stiffness elevation. All of this saying that the transmission of the mechanical forces is going through the cytoskeleton structure of the cells after our treatment. So in this project, for now, we've shown that MGO and AGE creation cause metabolic impairment in the adipocytes and in the mice model, cause changes in the ECM content and stiffness, activate YAP1, change intracellular structures as cytoskeleton and nuclear size, and probably all of those are part of the factors that help to the vicious cycle in this disease. So going back to the first chart, probably adipose tissue needs to be added to this diabetic complication that associated with AG is. And that's it. I want to thank my lab members and my PI, and for you to listen